Good morning. Thank you very much. I really want to thank the Academy and Eddie de Robertis in particular for inviting me to be here. I thought I would tell you a little bit today about the kind of work that my laboratory has been doing for the last 25 years now, and it has to do with a different way of doing biochemistry and biophysics, which involves doing it at the level of single molecules. We all are aware that most of the work that we do, the way we have done chemistry and biochemistry in the past, has been done by what I call the ensemble approach, where you have a large number of molecules A reacting with large number of molecules B to form a large number of molecules C. And you can play the movie for a second. Thank you. So this is the typical experiment we all saw in high school, where we can actually make two chemicals react, and we can follow that chemistry by following some general property, the color, for example. And we can follow the chemical reaction in this case, because this huge number of molecules are going from A, are combining A and B to produce C. And so these type of experiments are the type of experiments that with which most chemistry and biochemistry have been made in the past. There are a number of advantages to this so-called bulk approach or in multiplo type of approach, because, for example, they are very robust. The signals are proportional to the number of molecules. There are lots of molecules. The signal is robust. You can follow a macroscopic observable, for example, the color, in this case, of the solution, so you can follow the reaction in a continuous manner. But there are some disadvantages. You remember that all the processes that occur at the molecular level are stochastic, and therefore it is impossible to synchronize molecules. And as a result of that, when we follow the change of color, for example, of the solution, what you will find is that what we are really doing is to follow the average of the behavior of the molecules in the population. And so in this case, we are not really following what each molecule is doing. And in fact, you could actually claim that no molecule in the ensemble will follow the behavior of the average. In other words, some of the molecules will be reacting very fast, some of them slow, some of them in between. And the bottom line is that the average that we measure in this type of methods can be quite misleading. And here I like to use a poem by the Chilean poet Nicanor Parra, who actually expresses the idea of the perils of averaging. It says, hay dos panes, usted se come dos, yo ninguno, consumo promedio, un pan por persona. So averaging can be very misleading. And the same thing is true, of course, not only in society, but in science as well, when we think about molecules. The alternative is to actually do the experiments at the level of single molecules, what we call in Singulo Studies, where we follow the reactions one molecule at a time. And in the last 20 years, two crucial experiments actually have taken place. One is the ability that scientists have today to detect fluorescence from single molecules, and the other is the ability to physically grab and manipulate a single molecule. I will tell you more about the second part, which is the one that we have developed mostly in my laboratory. And so many years ago, 1991, 1992, we did this first experiment where we grab a single molecule of DNA, as is shown here. We put it between two beads, two plastic beads, which were microscopic beads, about 1 micron in diameter. And then we were able to pull one of the beads relative to the other, which was held atop a micropipette. And by applying force, we could actually determine how much force was needed in order to extend the molecule by a given amount. This is equivalent to study or understanding the elasticity of DNA. And this experiment, of course, I would be lying to you if I would tell you that we did this experiment with the idea that we could do what we can do today using this same technology. It was just that it was possible to do, we thought, and it would be interesting and fun, as the Americans call it, to do this type of experiments. Using this technology, we were able to show, for example, that if we plot the force in the y-axis of this plot versus the extension, in the x-axis, you see that, in fact, the behavior of DNA is quite nonlinear. It's a spring that is very nonlinear. The linear spring would be the dotted red line that I have actually indicated there, and that's the one that follows Hooke's law, who expressed his law in Latin by saying "Huc tensio sic vis," which means like the extension, so goes the force. The force is proportional to the extension. That's Hooke's law. But DNA doesn't follow Hooke's law. You can see it's much more complex spring, and it follows a nonlinear behavior. Now, why do we want to study single molecules? It's certainly easier to study many molecules at the same time, but there is, besides the point of being able to actually understanding how molecules carry out the reactions and not looking at averages, it turns out that many processes in the cell, such as chromosome replication and segregation, or DNA transcription, recombination, RNA translation, are often carried out by very, very few molecules in the cell. And we are interested, therefore, to know how do reactions happen in the cell at the single molecule level. What can they tell us about the mechanisms of the reaction? We also know that at the single molecule levels, reactions are very noisy due to the fact that the thermal fluctuations affect the behavior of the signal. So can we learn something about the molecules from those fluctuations? And the last point I want to make is that by grabbing molecules physically, mechanically, you can exert forces and torques. And it is possible to, in fact, for the first time, thanks to these methods, not only to measure the forces and torques that are generated in the course of a chemical reaction, but also, or a biochemical reaction, but also use those forces and torques externally to alter those biochemical reactions, and in the process, learn something about those reactions. So I'm going to tell you a story that actually we did using this type of methods, which involves a particular type of molecules in the cell, which are called ring ATPases. These are pentameric or hexameric rings of ATPases, molecules that can convert chemical energy into mechanical work. And I show you here, for example, the one that is at the base of many viruses whose function is to package the DNA inside the capsid of the virus. Then you can see also helicases that move in front of the RNA, the DNA polymerase during replication to open the strands of the DNA. There are DNA segregases that segregate the DNA in the cell in the left bottom corner. A famous ring ATPase is the ATP synthase, the molecule responsible for synthesizing ATP in the cell. And finally, as another example, I included there on the right-hand side, molecules that are ring ATPases whose function is to destroy proteins in the cell when the cell don't want that protein anymore. So in that case, it uses the ring ATPase to grab the polypeptide and pull the polypeptide through the ring and fold the protein and then digest the protein in a second chamber. So I'm going to tell you about one of these systems, but what you see here in all these examples that I have shown you is that these all involve the conversion of chemical energy in the form of hydrolysis of ATP into mechanical energy as a generation of work and force. Now, the mechanical paradigm in biology is not new. In fact, in 1666, Marcello Malpighi, who is considered the father of microscopic anatomy, wrote this book, De Viscerum Structura, where he said, the operative industry of nature is so prolific that machines will be eventually found not only unknown to us, but also unimaginable by our mind. This prescient statement from the man who was leading the revolution in biology in the 17th century is of great interest now historically, of course, because at the same time that this revolution was occurring in biology, thanks to the invention of the microscope, there was a parallel revolution that was occurring in physics, which were first Galileo and eventually Newton were casting the terms in which we could actually discuss the behavior of these machines precisely. And that revolution was thanks to the invention of the telescope. And so we see that both disciplines were actually breaking from the old mold and going into what we can consider the modern imagination in both disciplines, in both sciences, thanks to the invention of instruments that allowed scientists to see beyond what they could actually do with the naked eye. Today we know that many directional movements in the cell require the operation of tiny machine-like devices that operate as molecular motors. And molecular motors are nothing but enzymes that couple the catalysis of a downhill chemical reaction, that's the hydrolysis of ATP, to the performance of a mechanical task. And so they function as energy transducers, converting chemical free energy into mechanical work through the generation of force and torque. Today, I'm going to tell you about a motor that sits at the base of a virus. This bacteriophage, small virus that infects bacteria, is called Phe29, and it has a motor at the base. I'm going to show you here, yes, in blue, a ring of 5 ATPases. It's a homopentamer, and these 5 ATPases hydrolyze ATP and pump the DNA into the capsid of the virus. So these are the cylinders of the engine, if you want. And so we think of our motor as a 5-cylinder engine, right? 5 ATPases, identical ATPases. Okay, so it's possible now to follow the molecular trajectories by doing the following experiment. We actually grab one single bacteriophage capsid in one bead, and we attach it to the end of the DNA that has begun to be packaged to a bead that is in an optical trap. An optical trap is a technology that we use in which we use light to trap matter, in this case the beads, and we can actually follow the process of packaging by seeing how the motor actually brings the bead together, brings the beads together during the process of packaging. So if we can play the movie, you will see that as we have this setup now, there is the DNA, there is the virus, and now we add ATP. As soon as we add ATP, the motor begins to package the DNA. You can see a single motor. We are playing tag of war with it. We have enough resolution to see that the packaging is made out of periods of dwells and bursts, dwells and bursts. During the dwell, the motor doesn't move. During the burst, the motor packages the DNA. And so in the next slide here, you will see here are single molecule trajectories of motors packaging the DNA in time. No? So you see the length, the distance between the beads is plotted in the y-axis, and the time is in the x-axis. And you see that the distance is getting shorter because the packaging is proceeding, but all the trajectories are made out of dwells, where the motor doesn't package, and bursts, where the motor actually packages exactly by 10 base pairs. So after every dwell, there is a 10 base pairs burst of packaging, and then again the motor rests, and then another 10 base pairs, and so on and so forth. But we have enough resolution in our experiment to actually see how is that each one of these 10 base pairs bursts are made up. And in fact, that's where our first surprise came. I told you that there are 5 ATPases, and I'm telling you that the burst of packaging is 10 base pairs. So the natural thing is to think that each ATPase packages 2 base pairs of the DNA. But in fact, what we found is that we see that a 10 base pair burst is made out of 4 2.5 base pairs. So 4 times 2.5 still is 10. But that means immediately that one of the ATPases, of the 5 identical ATPases, is not doing any work. We call it the PI, the professor. The other 4 are the graduate students. So 4 graduate students doing the work, 1 professor just telling them what to do, right? And so this is quite interesting because, in fact, they are otherwise identical ATPases. So that was a very interesting observation. And I just tell you quickly what we know about the motor. One is that it is a very powerful motor. It can generate out to 70 piconewtons of force. I remind you that a Newton is the weight of an apple on the surface of the Earth, approximately, right? So we are measuring in 10 to the minus 12 newtons, that's the piconewton. And 70 piconewtons is strong because myosin, which is the muscle protein, the motor in our muscles, can generate only between 3 and 5 piconewtons of force. So this is very powerful motor. The other thing is that the moment in which the power stroke is produced in the motor coincides with the release of phosphate from the ATP after hydrolysis has occurred. The other thing is that the ATPases, all 5 of them, are highly coordinated. If you stop one of them, then the whole motor stops. All of them has to be working in order for them to actually work well. And I already told you that packaging occurs in cycles of dwells and bursts, and the bursts are 10 base pairs in size, 4 2.5 base pairs. And we found, in fact, that every 10 base pairs, the motor has to contact one of the DNA phosphates in the DNA in order to be able to reset itself and be able to package the next 10 base pairs. So we did this by converting DNA into DNA phosphonate. That's the detail, by essentially eliminating the electric charge from the DNA, locally confused the motor tremendously because the motor needs to make that electrostatic contact with the phosphate every 10 base pairs. And that contact seems to be important both for holding onto the DNA, but also for regulatory functions. And what I just told you, one subunit is special in the sense that it doesn't work and must do something else. Okay, so here is how we look at the dwells and bursts that I was telling you. During the dwell, we know that the motor binds ATP, and during the burst, the motor packages the DNA. When it hydrolyzes ATP, releases the phosphate, and then does the power stroke of 2.5 base pairs. 4 of them. 4 of them times 2.5 is the 10 base pairs. Okay, that's what we knew. And we wanted to know other things. For example, where is that hydrolysis occurs? And we can think of 3 scenarios. One is that the hydrolysis occurs during the dwell. So you get binding of ATP, hydrolysis, binding, hydrolysis, binding, hydrolysis. In other words, hydrolysis is interlaced with binding, scenario 1. Scenario 2 is that it happens in the dwell as well, but segregated from ATP binding. So you get binding, 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 and then hydrolysis, right? Or the third scenario is that the hydrolysis actually occurs during the burst. Just before releasing the phosphate, the ATP is hydrolyzed, and then the motor actually power strokes. We can actually find out which one of these scenarios is the one appropriate if we use a molecule that looks like ATP, but that the motor cannot hydrolyze. It's an analog of ATP that is not hydrolyzable for the motor, and determine where is that we stop the motor. So suppose that this molecule is called ATP gamma S. Imagine that actually is the first scenario. Since hydrolysis occurs in the dwell, the motor will stop at the dwell. And the distance between the dwell, the last dwell, and the pause produced by the ATP gamma S is exactly 10 base pairs, right? Because it's the last burst. If it is the second scenario, again, the motor will stop and create a pause at the dwell, and then the distance between that pause and the last dwell will be again 10 base pairs. But if it is the third scenario, the motor could stop here, or could stop here, or could stop here, or could stop here, right? And depending on where it stops, because it's occurring during the burst, then the distance between the last dwell and the pause will be either 2.5, 5, 7.5, or 10 base pairs. So let's see what happens when we are ATP gamma S. We are ATP gamma S, and the motor, in fact, pauses. The motor is packaging. You see the distance. In the y-axis is the distance, and in the x-axis is time. And you see 3 different examples there on the left, where the motor pauses, and then it actually eventually continues packaging, because presumably the ATP gamma S has left the motor, and a regular ATP now has come and bound to the motor, which can be hydrolyzed. But notice something interesting. The motor sometimes just pauses one time and leaves. But sometimes it pauses, then it makes 10 base pairs and leaves, then pause 10 base pairs, pause 10 base pairs, pause 10, and then eventually the motor continues packaging. What's going on? It turns out that we can show that all this complex behavior happens when just one single ATP gamma S is bound to the motor. One single ATP gamma S is bound to the motor. And so what this means is the following. Notice you have pause for a long time. This is not a dwell. The dwell is only 80 milliseconds long. These are seconds, 4 seconds, 6 seconds, pausing, waiting. And then eventually they do 10 base pairs, and then pause again. Then 10 base pairs, then pause again. It's almost as if the motor is stuttering, right? Okay, so to make a long story short, what we think is happening here is that the one subunit that has bound the ATP gamma S is the special subunit, whose function is to regulate the motor. And in order to regulate the motor, it needs to actually hydrolyze its ATP. And because it's bound to the non-hydrolyzable ATP, it cannot send the signal to the other 4 to do it. And eventually they do it spontaneously, just because thermally they cross a barrier and spontaneously begin to hydrolyze and create the burst of 10 base pairs. That's the interpretation of this experiment. And I will basically like you to concentrate in the last sentence here. The motor can still function with one analog bound, retaining the coordination of the other 4 subunits, albeit very slowly, because it's a long pause, 10 base pairs, long pause, 10 base pairs, etc. So, which one of these, where is that hydrolysis occurs? Remember I told you, by looking at the distance between the last dwell and the pause, we can tell whether it's the first scenario, the second, or the third. It turns out that sometimes we see that the distance between the last dwell and the pause is 2.5 base pairs, sometimes 5 base pairs, sometimes 7.5 base pairs, sometimes 10 base pairs. So that clearly indicates that the hydrolysis of ATP by the motor occurs during the burst, right? Which is the prediction from this scenario. Well, you may ask also, for example, where is ADP release from the motor? I will not bore you with how we actually determine that. By using similar approaches, it's possible to show that ADP is released during the dwell, and in fact in an interlaced fashion with the binding of ATP. And for those of you who are biochemists, I'll remind you that what this scenario predicts is that since one ATP has to leave in order for one ATP bind, and one has to leave for another one to bind, and so on and so forth, at any given time, the motor only has one ATP binding site available, right? Open to be bound by ATP. So the motor behaves not as a cooperative machine, say hemoglobin, but as myoglobin, because it has only one binding site. And that's the behavior of this motor. So we were able to use that argument to actually show that the motor, the release of ATP is interlaced with that of ATP binding. Well, so what is the role of the 5th subunit? I told you that actually under conditions in which the 5th subunit has bound ATP gamma S, it cannot actually direct, the motor behaves like disconcerted. It's waiting there and waiting until eventually, spontaneously, hydrolyzes and packages 10 base pairs, and again waits and 10 base pairs, and so forth. So we believe that what is happening is indeed that this subunit, which we call the 5th subunit or the special subunit, is one that has the function of directing the motor and coordinating the packaging during the cycle of the motor. And when it cannot send that signal, the other subunits are of course unable to do the packaging in a timely manner. How many minutes do I have?
47 seconds.
47 seconds. Okay, so I will tell you now what event breaks the symmetry of the motor. There are 5 identical subunits, and one of them becomes different. It turns out that we think that the reason they are different is the following. Here you can see experiment we did where we ask a different question, but help us answer that question. And is, does the motor rotate the DNA while packaging? And in order to do this, I'd like to show you this video where you see that if you put a tiny little bit on the side while the packaging is proceeding, you see that that little bit is rotating. indicating that in fact the motor packages the DNA, but not only exerts force, but rotates the DNA as it actually packaging the DNA. And why is this important, and why is that this actually help us answer the question of the... It's important because when you ask the question, how much rotation it occurs in the DNA, it's only 1.5 degrees per base pair. And that 1.5 degrees per base pair indicates that the motor is really contacting, every 10 base pairs is rotating the motor, the DNA by 1.5 degrees per base pair, or 15 degrees per 10 base pairs. And I'd like to show you a movie now at the end. It's a very simple movie, but it actually tells the story of the whole, what we have learned about the motor. At the end of packaging, at the end of the burst, you have all the molecule has ADP, and then the special subunit releases ATP. Binding of ATP produces ADP release of the next one. Binding of ATP produces ADP. Binding produces ADP. And now the special subunit is the first one hydrolyzing this ATP and telling the other subunits, hydrolyze and 2.5, 2.5, 2.5, 2.5, and then the motor rotates the DNA by 14 degrees, 15 degrees, and then again the release of ADP and the exchange between ATP and ADP. So this is a machine that actually works almost like a clockwork, and it's actually is an example of the many machines in the cell that actually carry out this type of work. And it's possible now to study how these motors work because we can use this single molecule approach that allows us to see the mechanism. Thank you.
Thank you very much.